Hi, everyone, and welcome to the fifth meeting of the Ask the Researcher Mpa Monitoring Series. My name is Lindsay with the Ocean Protection Council, and I'll be facilitating this webinar alongside a few other OPC folks. Tova's on the line and Elise, our CDFW partners, Steve Wirtz and Rochelle with Strategic Earth. This webinar series was designed as an opportunity for interested members of the public to interact directly with the researchers involved in California's Mpa long-term monitoring projects. Our goal in facilitating this 8-part series is to support broad understanding of the research that is informing the Mpa 2022 Decadal Management Review. Last fall, we hosted 4 community meetings to inform the public about the Decadal Review and to hear your perspectives and priorities on Mpa management. A consistent piece of feedback we heard was to let folks hear directly from our researchers to help them better understand the science that is informing the review. But before we begin, I like to go over a few technical things and meeting agreements that will help promote a safe and respectful meeting for everybody. So first, if you haven't noticed, this webinar is being recorded. Can we back up one more slide really quick? This webinar is being recorded, so just be mindful of that. And sorry, we can back up here. I have these. There we go. If you have any technical difficulties viewing the screen or calling in, please email Rochelle with Strategic Earth. She'll drop her email in the chat there or message her directly. Remember to unmute to speak, so we'll call on you during the Q&A as we see your names pop up in the raise hand, or you can press *6 if you're joining via phone. It'd be really helpful if folks that are participating via Zoom on your computers to change your name to help identify yourself and other participants. So to do that, go ahead and click Participants in the meeting controls, locate your name and click More, and then choose Rename. Please enter your full name and some sort of identifier so we can know who you are. This will be closed captioned, and so if you want to turn on subtitles, click CC in the meeting controls. So one more thing before we get going, we have some meeting agreements. So if you could all remember to please put yourself on mute when you're not speaking, that'd be really helpful. Please listen for understanding and acknowledge and seek clarification from others. Please openly discuss issues with others who hold diverse views and approach discussions from a place of diversity and inclusion, considering all voices and perspectives. Please keep your comments concise and focus on the topic at hand. Remember that personal attacks and offensive language will not be tolerated. And connect with facilitators or agencies to talk through any questions or concerns you might have. We ask that everybody abide by these agreements. And if you're unable to, we will invite you to leave today's meeting and watch the recording that will be posted in the coming days. You're also encouraged to email your input to CDFW at the address that's being dropped in the chat. So without further ado, we're very fortunate today to be joined by Dr. Scott Hamilton from Moss Landing Marine Laboratories, who will be giving us a brief presentation on his work on the California Collaborative Fisheries Research Program. After the presentation, we'll have time to hear questions from the audience. Now I'll turn it over to Dr. Hamilton.
All right, hopefully everyone can see the slides. So yeah, I'm very excited. All good?
Yep, all good.
Perfect. All right, well, yeah, I'm really excited to talk to everyone today about the results of the Mpa monitoring that we've done with the California Collaborative Fisheries Research Program, or CCFRP. So I'm the lead PI of the program, and I've been doing this role for about 4 years now. But the program was started by Rick Starr of Moss Landing Marine Laboratories and Dean Wendt at Cal Poly San Luis Obispo, probably back in about 2005 with the first sampling we did in 2007 through this process that involved stakeholders and volunteer anglers and sport fishing captains and resource agencies. And we expanded the program in 2017 to be statewide to include 6 institutions. And so you can see the names of some of the other PIs that are part of the program. So a little outline of the talk, just a quick background about CCFRP and sort of an introduction to the Mpas. I'll tell you briefly about the methods that we use to monitor the Mpas, which are with hook and line fishing techniques. Then we'll dig into the results. So we'll talk for a little bit about some of the long-term results. We have a 15-year time series from 4 Central Coast Mpas, and then results from the statewide program, which is a 5-year time series now, extending from Northern to Southern California. We'll then talk a little bit about results from our tag recapture study, where we can look at fish movements and whether fish are moving across the Mpa boundaries or spilling over. And then results of a survey that we did recently with our volunteer anglers to ask questions about their perceptions of the Mpas, fisheries management, and conservation. A little bit of discussion, and then hopefully quite a bit of time we can answer some of your questions. So CCFRP, what we are, we're a community science program. We're a fishery independent catch and release research program, meaning we go out there and we fish in a very scientific and standardized way. And our program combines the expertise and the ideas of the fishing community. All the people that come out and help us catch fish and collect the data for us are volunteer fishermen from a whole variety of backgrounds and experience levels. We work really closely with the sport fishing industry and captains and crew that help us go out and fish and help us to design the methods that we use. The academic scientists, we helped put together the experimental design and we run the logistics of the program. And then we have a lot of involvement from resource managers and other agencies. And you can see there on the bottom, a number of the groups that make up CCFRP. And the program we developed conducts scientifically rigorous data collection and analyses, both for Mpa monitoring as well as for fisheries management. So a lot of the data we've collected has been used in the stock assessments of 7 nearshore species, especially ones that are data limited. And then we try to do a lot of outreach and education with the angling community to show our results and to get feedback and to improve the program. So we're doing all this monitoring because of the Marine Life Protection Act, right? So the MLPA, this was a law that was passed in 1999 that mandated the creation of a network of marine protected areas with the goals of protecting the diversity and the ecosystem function throughout the state of California. It was implemented in a number of regions, starting in 2007 in Central California, moving up to the North Central Coast, down to Southern California, and then back up to Northern California, finishing around 2012 through a process that involved a whole variety of stakeholders providing input and the best available scientific advice and guidance on the sizing and spacing of the Mpas and things like that. Currently, there's 124 Mpas in California that cover about 16% of state waters. And if you see there on the map that I tried to piece together, the red spots and the blue spots, the red ones are state marine reserves that are no take of any kind, whereas the blue ones are state marine conservation areas that allow the limited take of certain species depending on the particular location. And we focused all of our work mainly in those state marine reserves. So for the decadal review of the Mpas, we put together a big report, addressed, 20 or 30 or more questions, and I've tried to distill those down here into kind of the 5 key ones we'll talk about today. So kind of the first question we'll look at is, are there differences in the abundance, size structure, and biomass of fishes inside and outside of Mpas, and have they changed over time? Secondly, do the Mpas provide resistance or resilience to climate disturbances? And looking at the response to a big marine heat wave. How do factors such as fishing pressure, Mpa area, Mpa age, or geographic location of the Mpas affect the strength of the responses that we see? Number 4, is there evidence of spillover of fishes from the Mpas, right? Or are the Mpas large enough to contain the home range of a number of fish species? And then lastly, how have perceptions of the Mpas changed for volunteer anglers that participate in our program? So here's the California coast. We break it down into these 3 bioregions: Northern California, Central California, and Southern California, each with a different complement of Mpas. And we have 2 of our academic institutions situated in each one of those regions: Humboldt and Bodega Marine Lab up north, Moss Landing Marine Labs and Cal Poly in Central California, and UC Santa Barbara and Scripps Institution of Oceanography, UC San Diego in the south. On the right there, you can see the map of the state with the Mpas that we sample. We can't sample in all the Mpas, but we chose a subset of the high priority Mpas. Each institution has 2 Mpas that they sample in, and an associated reference site that has similar habitat as possible to the Mpa. Going down the coast, Humboldt samples in South Cape, Mendocino, and 10 Mile. Then occasionally there's an odd site that has maybe been sampled a few years that's not part of the core program, like Trinidad. Bodega Marine Lab samples at Stewart's Point and Bodega Head. Moss Landing Marine Labs, we sample at Año Nuevo and Point Lobos. Cal Poly samples at Piedras Blancas and Point Buchan. UC Santa Barbara samples at Carrington Point and Anacapa Island out of the Channel Islands. And Scripps samples at Swamis and South La Jolla. So there's a paired marine reserve and reference site that we sample, and we go to each site multiple times a year. So the methods will zoom in on the Central California coast to give you a sense for what we do. So there's sites like Año Nuevo, Point Lobos, then further south, Piedras Blancas, and Point Buchan. Each one of these areas you see outlined in kind of the pink color, that's the boundaries of the MPA. Then these red squares are the MPA sites that we conduct our hook and line surveys in. The blue squares are the reference sites, again, that were chosen to have similar habitat to the MPA sites. And each one of those is a fixed 500 by 500 meter grid cell. We randomly choose these to go fishing every day when we go out sampling. And again, they're chosen to be in sort of in rocky habitat and in an appropriate depth zone that we can have high survivorship of the fish when we release them. And so when we visit a fishing cell, now the sport fishing captains have a lot of latitude for how we fish, but we aim to fish for 45 minutes in each cell. That's broken down into 3 15-minute drifts, like you can see inside that square. And the data that we collect when we're out there is we collect the start and stop times of the drift, GPS coordinates, so we know where our drift went over. We keep track of all the anglers on the boat, which fish they're catching, what species. We measure the lengths of each fish in this measuring board. We record the condition of the fish in case they have any damage from the hook. If they're in good condition, we will tag them like you can see in that picture on the lower right. And then we release all the fish. And for many of the fishes, we use descending devices to help take them back down to the depth we collected them and to release them, which really improves their survivorship. So a summary of our program from 2007 to 2021, we've sampled 690 days at sea. We've worked with 31 sport fishing boats and 53 captains. We've had over 1,800 individual volunteer anglers come out. And so again, they're really the core of the program that helped us collect all the data. We've collected over 200,000 fishes of 95 species, and we've tagged and released about 65,000 of those fishes. So let's get into some of the results. So this first part is about the long-term monitoring from the Central California MPAs, where we have 15 years of data. So how we measure abundance is measured as a catch per unit effort, or a catch per angler hour. And so what we do is we add up for any species for all the fish we caught. We add up the numbers of fish that we've caught. We divide it by the time that was spent fishing on that drift or in that cell. Multiply that by the number of anglers that are on board. Sometimes we have more or less anglers fishing. And we subtract out the amount of time people are not fishing, which can be because their gear gets tangled, which always happens to me, or they take a break or for whatever reason. So we have a very accurate estimate of the actual amount of time that all the anglers are spending catching fish. And the assumption is if you're catching more fish for the same amount of effort, there's likely more fish in there. So this is a relative measure of abundance. And so here are some of the results. So over on the left shows you the average Cpue, or catch rates of fish inside the MPAs. They're the red points. And then the reference areas open to fishing or the blue points over time from 2007 to 2020 on the bottom x-axis. And then the lines that you're seeing are the best fit linear trends showing what the catch rates have been doing over time. So hopefully you'll notice there over on the left that in general for the first about 5 to 7 years of the program, there weren't really big differences in catch rates inside or outside of the MPAs. But after about 2014, there was a big difference. Catch rates really increased inside the MPAs. They also increased in the reference areas, likely due to some beneficial environmental conditions, but they increased much faster inside those MPAs that were closed to fishing. If you look over on the right, you can see what's happening in the 4 sites individually. Hopefully you'll see that Point Lobos and Point Buchan, right, there's much bigger differences in the catch rates inside the MPAs than out. Catch rates are pretty flat outside the MPAs, but they went up really quick inside the MPAs, especially after 2015. Piedras Blancas, they're going up both inside and outside of the MPAs, but faster than the MPAs. And then in Año Nuevo, we actually have higher catch rates outside of the MPAs, but they're both going up over time, again, reflecting the environment. We also go out there and we measure the lengths of all the fishes that we catch using that measuring board. And so these plots here show you for the 10 most common species we encounter, what's the average length of the fish over time. And I'll just highlight here in green some of the fishes that show some really strong patterns, where hopefully you'll see that the red points, the MPA points, are higher than the blue points, the reference site points. showing that fish are larger inside the MPAs through a lot of the time series. You see that for lingcod, for copper rockfish, for vermilion rockfish, gophers, black rockfish, and olive rockfish. Now we use this size information along with that Cpue, that catch information, to actually come up with another metric that we use, which is called the biomass per unit effort. So a metric of biomass uses the weight of all the fish, and we use the lengths that we measure and publish relationships that can convert lengths to weights to estimate the weight of every fish we've caught. And then we add up all those weights to give us a new metric that's the kilograms of fish that are caught per angler fishing per hour. And often biomass is a more sensitive metric of MPA responses because it incorporates those two pieces of information, both the length of the fish and the abundance of the fish, into a single metric. So most of the rest of the talk will show you biomass responses in the MPAs. So here's those same plots, but now using biomass instead of catch rates. They look very similar to what you saw before, except for the differences are magnified, because often we're catching bigger fish in the MPAs. So now we're seeing even higher biomass inside the MPAs compared to the reference sites. And the biomass is going up both in and out, but it's going up much faster inside the MPAs. And if you look at the 4 individual sites, once again, Point Lobos and Point Buchan, you see those big differences. Biomass is pretty much flat outside the MPAs, but it's gone up pretty quickly inside the MPAs. Peters Blancas, now there's a bigger difference in those trends. It's going up faster in the MPAs than it was before. And Año Nuevo, if you remember a couple slides back, the blue line used to be above the red line, higher catch rates outside of the MPA, but now the red line is above the blue line. And so we're catching similar or slightly higher biomass inside the MPA, because even though there's fewer fish, they're larger in size on average. Now, if we look at individual species, we see some interesting trends over time. Overall, 73% of the species have higher biomass inside the MPAs. The ones that don't are generally ones that we encounter fairly infrequently. But here shows the 10 most common species that we encounter. And I'll highlight a few of the interesting trends that jump out. So for species like blue rockfish, olive rockfish, copper rockfish, vermilions, and gophers, we see a trend where you see these big differences in the MPAs, especially in the last 5 years, the most recent 5 years in the time series. That's when those differences really occurred. And in general, the catch rates outside the MPAs are pretty stable, but they really increased inside the MPAs in the more recent years. We also see some other patterns here like kelp rockfish, lingcod, canary rockfish, or yellowtail rockfish, where you see a peak in that biomass right around 2014, 2015. And then for many of those species, the biomass declined. But when we were at that peak, it was higher inside the MPAs than in the reference sites. And I'll show you a little bit later how this might be related to some of the climate impacts that we've had. And then there's a species like black rockfish where we actually have higher biomass outside the reserves, mainly due to the Año Nuevo Reserve, where we catch a lot of black rockfish in the reference site. Right, so now we're going to talk about some of the responses of the fish community to the marine heat wave. So there was a big marine heat wave off the whole West Coast, 2014 to 2015. It's also affectionately called the blob. And that warm blob just sort of sat on the coast for multiple years. These plots here show you sea surface temperature anomalies, so sort of differences from the average temperatures in a couple different time periods. On the left, September 2012, before the heat wave, you can see those bluish and yellow colors suggesting the waters were kind of cool, about normal that time period. September 2015, right in the middle of the heat wave, everything's red because the waters are much warmer than normal. They're about 3 degrees Celsius hotter than they normally are. And then after the heat wave there in 2018, they've returned back to kind of those normal like yellow and blue colors. So how did the fish community respond? Well, one way we looked at this was by measuring it with a metric of biodiversity, which is calculated by how many species are there, as well as sort of their relative abundances. And you'll see here that the gray area on that plot shows the timing of the heat wave. So before the heat wave, the red points are above the blue points. So the MPAs had higher diversity than the reference sites. Then during the heat wave, you'll see both the MPAs and the reference sites, the diversity kind of collapsed. So the MPAs did not really resist the effects of the heat wave in terms of how the community changed. But then you'll see the red points increase faster than the blue points afterwards. So the MPAs increased in diversity more rapidly, which is a sign that they were more resilient. They kind of bounced back more quickly than the reference sites. And some of those changes we observed, it was mainly a reorganization of the fish community. A few new species showed up from Southern California, like ocean whitefish and California sheephead, but primarily the species that were there really changed in abundance. And so these plots on the left show the MPAs before, during, and after the heat wave. And this is the percent or the proportion of the community that each fish makes up, each species makes up. And then on the right is the reference sites. And I'll highlight here for you. You can see black rockfish, they became less common in the community as the heat wave progressed. Whereas a species like the blue rockfish here, you see those bars get bigger, they became a much bigger player in the community. And then a few other species, as you can see, have changed their relative abundance. So the community shifted and a few new species showed up. This is that same plot I showed earlier of the changes in biomass over time, but now we've highlighted the timing of the heat wave, and you'll see, those different patterns that we were talking about are mainly related to the timing of the heat wave. So the kelp rockfish and the lingcod and the yellowtail rockfish, right, they're peaking right during the middle of the heat wave and then declining, probably in response to that heat wave. Whereas species like the blue rockfish, the coppers, and the gophers, olives and vermilions, you see them really increase right after the heat wave, potentially due to the environmental conditions that influenced the arrival of their offspring. All right, one other metric that we calculate using the biomass values is called a response ratio. And for this, we divide the biomass inside the MPA by the reference site, and we take the log of that, the natural log of that value. And it gives us a new metric where a value of 0 means there's the same biomass inside and the outside of the MPA. If the value is positive, there's more fish inside the MPA. If the value is negative, it means there's more fish outside the MPA. And so we've looked at that over time until you see right around 2007 when we started sampling, we're at a value right close to 0, suggesting there's similar biomass inside and the outside of the MPAs when they were first established. And then that biomass response increases over time, meaning that the biomass is increasing faster inside the MPAs than it is in the reference sites. So this is a good sign the MPAs are working. We would expect, again, the biomass to really diverge between the MPAs and the reference sites over time. If we look at our 4 sites, again, we'll see that Point Lobos and Point Buchan are showing really strong responses, where the differences between the MPAs and reference sites are increasing over time. Piedras Blancas is a bit weaker, and Año Nuevo is really no different from 0. And so we are really curious what factors might explain some of these differences in the strength of the MPA responses that we're seeing across these Central California sites. One of the factors that turned out to be very important was the amount of fishing effort that's happening outside the MPA. And so for this, we used information that we collect from all those MPA cells, right, where there should be no fishing happening. And then these reference site cells that are open to fishing. And we used information from California Department of Fish and Wildlife. They have information from these microblocks, these gray squares you see in the background, 1 by 1 nautical mile blocks, where they record information from angler surveys about how many people are going and fishing in these areas. And so we use that microblock fishing effort data for the same locations where we have our reference cells to ask whether or not these MPA responses change depending on how much fishing effort you have outside the reserve. And so this is what we saw. Fishing effort here is on the x-axis. The strength of the MPA responses is on the y-axis, again, which is the difference between the MPA and its associated reference site. And we see this positive response. So in places where there's much higher fishing effort, there's a bigger difference between the MPA and the reference site, with much higher biomass in the MPA. Places where there's lower fishing effort, the MPA and the reference site are more similar, which you would expect if there's not much harvest outside the MPA. In those 2 sites where we saw the weaker responses, that's Año Nuevo and Piedras Blancas, which tend to be a bit further from port. The 2 sites where we saw those strong responses to the MPAs were Point Buchan and Point Lobos, where there's higher levels of fishing effort. All right, so let's transition to the statewide MPA results. So here now we have 12 MPAs and their associated reference sites. So you can see that graph over there on the left, going from north to south, from Cape Mendocino down to South La Jolla. At every MPA we survey, you'll see those red bars are greater than the blue bars. So we're catching a higher biomass of fish inside the MPAs compared to the reference sites. But you do see there's some strong geographic variability where we catch lower biomass, particularly in Southern California, and typically higher, in Northern and Central California. In each region we go to, over 70% of the fish species have higher biomass inside the MPAs. And so we've gone to try to look at what factors might explain the differences in those responses across the whole state. And these are the ones we've looked at, so I'll highlight them. So the first is Mpa area. So as you go from small reserves to large reserves, you see that we see a stronger Mpa response in the large reserves, potentially because they're protecting a larger proportion of the community or protecting the home ranges better of those species. So the Mpa's differences are much larger in those large reserves. The location on the coast is also important. So here's latitude going from south to north. We see stronger Mpa responses in the south in Southern California than in the north. I think this is probably partly due to the fact there's more people in Southern California. There's a lot more people out there fishing. So you get a bigger difference between the protected areas and the areas open to fishing. Also potentially due to the species. Those in Southern California tend to grow faster. They tend to show up more predictably as offspring, so they might be able to respond more quickly. Down here on the lower left, this is another metric of fishing pressure that we've used that works across the state. And again, we see a positive response. In the sites that have higher fishing pressure, there's a bigger difference between the Mpas and the reference sites. Places where there's lower fishing pressure, the Mpas and reference sites are more similar to each other. And then lastly, Mpa age is also important. The older the Mpa is, the stronger the difference we see between the Mpas and the reference sites, which makes sense, right? Because it's going to take some time for those differences to accumulate. All right, so looking at our tag or capture data, we're interested in whether or not there's evidence of spillover from the Mpas, right? Are fish moving from those Mpas into those reference sites that are open to fishing? And we can use our tag or capture data to answer some of these questions. Right, so fish that are in good condition, we'll tag them with a little T-bar tag, a little spaghetti tag, and release them. And when they're recaptured either by us or other anglers, there's a phone number on there. People can call in and give us information about the fish, where they caught it, how big it is, and we can use that information to get information on movements and growth. And these tags can be on the fish for a long time. We've caught fish 7 or 8 years after they've been tagged, and they still have the tag on them. And we put out these flyers to try to inform people that we would like to have those tag returns, and we provide rewards for it, and showing some images there of kind of how foul the tags can get. And then when people call in those recaptures, we usually send them a flyer, giving them information about the fish, where it was tagged, how big it was, how much it grew, and some natural history and life history information about the fish. And we can use those recaptures and the distances between where they were tagged and recaptured to get information on their net movements and potential spillover. So on the left, you'll see a histogram that shows, in general, the net movements of all the fishes that we've recaptured. You'll see most of the fishes aren't moving very far at all, maybe, less than 1 kilometer or so from the place that they were originally tagged. But some fishes move longer distances, 5, 10, or even over 100 kilometers. So I'll give you a few examples. Here's gopher rockfish, right? Many of them were recaptured in the same cell where they were released, so they're not moving very far at all. But a few individuals moved between cells, mainly on the order of a couple kilometers at most. But the majority of these individuals don't move very far at all. Copper rockfish are an interesting species. They kind of fall on either sides of the spectrum. Many of the copper rockfish we catch are recaptured in the same place they were released, even years later. But some individuals decide to move, and those that have moved on the scales of tens, even a couple 100 kilometers along the coast, like you can see here with some of these movements. Here's black rockfish. They're this end of the continuum. They typically are highly mobile and move pretty far. We're usually catching immature individuals, and they're almost always recaptured off in Northern California and Oregon. I think they're moving up that way to migrate, or they're migrating up there for reproduction purposes as they get older. So for the fishes that we've tagged in the Mpas, we've tagged about 25,000. We've recaptured about 136 of those, so it's a pretty low recapture rate. But remember, we're not out there fishing very often, only a couple days a year. And the population sizes of these fish are quite large. But for those fish that were tagged in an Mpa and showed up somewhere else, 17% of them spilled over to an area that was open to fishing, which means, 83% stayed inside those Mpas. So if we look at these plots, we can see for our 4 Central California sites, the proportion of fishes that either stayed in the Mpa in blue, which are most of the fish, or those that spilled over and moved across the boundary, which you can see in red. So there's a bit of variability from site to site. I'll just point out Año Nuevo and Piedras Blancas. Again, these are the two sites that showed the weakest MPA responses. Part of that was due to the fact that fishing effort's pretty low, but also it could be partly due to the fact that many of these fishes are moving across the boundary and redistributing themselves. And in Año Nuevo, it's mainly the black rockfish. In Piedras Blancas, it's the gopher rockfish that were doing a lot of the movements. This is hot off the press. We've started to do some other analyses looking at evidence for fishing the line behavior. And for that, we've calculated the distance between every one of those cells we fish and the nearest MPA boundary. So in this plot, negative values are going into the MPA, whereas positive values are increasing distances away from the boundary of any of the MPAs. And here are biomasses on the y-axis. And if you follow the red line, which is the best fit trend, you'll see you have the highest biomass in the center of the MPA, tends to decline as you go towards the boundary and across the boundary, with this prominent dip about 1 to 2 kilometers past the boundary of the MPA, and then rises up again. Theory and other studies have shown that this is the exact pattern we would expect to see if people are tending to go and fishing near the boundaries of MPAs. It's also evidence the MPAs are providing this spillover benefit, right? People are going there likely because they're being able to catch some of these larger fish. All right, and then the last bit of results I wanted to tell you about are the statewide angler survey we did. So we put a survey out to all of our volunteer anglers, about 1,400 at the time the survey went out. We got 262 responses, or about a 19% response rate, which is pretty good for a survey. And we asked people about 30 different questions about their perceptions of MPAs and their attitudes towards fisheries management and conservation, as well as some demographic information. You can see there from the map on the right, we got responses from zip codes throughout the state of California, mainly coastal, but sometimes inland, and from north to south. A few of the highlights, we asked people whether their opinions of the MPAs changed after they volunteered with CCFRP. So what was their opinion of MPAs before they volunteered with us? Was it positive, somewhat positive, neutral, somewhat negative, negative or no opinion? And that's in the white bars on the left. And then what was their opinion after volunteering with us? And those are the black bars. And then we calculated the difference. That's the plot over there on the right, showing you that there was a big increase in the positive perceptions of MPAs after people volunteered with our program, mainly because they were changing their opinion from all the other categories. We're also curious whether the longer people volunteered with us, whether their opinions changed more. And so we see that here in this plot. And at the orange shows people that volunteer every year with CCFRP had a stronger change in their positive perceptions of MPAs than those that volunteered much less frequently with the program. And part of the reason we think that people are having more positive views of MPAs is probably because of what they're seeing when they're coming out with us and fishing inside these MPAs. So we asked people, do you observe differences in the abundance of fishes, the diversity and the size of fishes when you come in and you're able to fish inside and outside of the MPAs? And the anglers reported that they catch, more fish inside the MPAs. So those are the taller bars. They catch a greater diversity of species, and they catch larger sizes inside the MPAs. So we think a lot of this has to do with the fact that you're able to really see for yourself. Right? I'm a scientist, I can show you the data, but really when you come and see for yourself these changes and that you're catching more fish and larger fish inside the MPAs, it really brings it home.
And Scott, just a gentle check-in. I know you're almost done, but just 5 more minutes, and then we'd love to open it up for questions.
Yeah, this is basically it, just to sum it all up and provide people with reminders of what I talked about, or in case they missed it. So the key take-home points are that the MPAs appear to be working pretty well, especially in the habitat that we are working in. We're finding the fish are larger in body size, they're more abundant, they're higher in biomass, and basically all the MPAs that we sampled. We found that the MPAs did not resist the effects of the marine heat wave, which depressed diversity and resulted in these changes in the community. However, we saw the MPAs were more resilient, such that they bounced back more rapidly than the fished areas. We found that fishing pressure, Mpa area, Mpa age, and the location on the coast help explain differences in the strength of the Mpa responses we're seeing across the network, why there's bigger differences in some places than others. Tagger capture data, right? We showed there's definitely some evidence of spillover. Fish are moving across the boundaries, but generally the majority of the fishes are staying inside those MPAs for pretty extensive periods. So it appears the MPAs are appropriately sized to encompass the home ranges of many of these species. Not all of them, but many of them. And lastly, hopefully we've shown you that our program has, right, there's strong power in this collaborative research, right, involving people, right, from many different backgrounds, conducting rigorous evaluations of MPAs. And hopefully as well that our outreach and education to the fishing community have produced some tangible benefits in terms of increasingly positive opinions of the MPAs. Right, so with that, you can find us on social media. I don't do it, but all my technicians and the students here do. So YouTube, Facebook, Instagram, and Twitter. Right, I'd like to thank all the volunteer anglers and the fishing captains and all of our partners that have helped to collect all this data and contribute to the results that we found. There's the email there at the top and our contact information. Our website is down below. If you're interested in coming out fishing with us or getting involved, please reach out. And yeah, with that, happy to take any questions from people.
Thank you so much, Dr. Hamilton. That was an incredible presentation with a ton of results packed in there. So I hope people have lots of questions. We have about 20 minutes to answer questions from you all. So we ask that questions be relevant to the topics at hand, and we leave space for everyone to have an opportunity to speak. If you're on the phone, you can hit *3. And if you're in the Zoom on your computer, you can just raise your hand and we'll call on you and unmute you when it's your turn. You're also welcome to ask questions in the chat, and we'll include those in the queue of questions for Dr. Hamilton. So I don't see any quite yet. We'll give it a minute. And if not, I will ask my first question. Oh, great. I see one from Dick Ogg. Go ahead, Dick, if you can unmute yourself.
Okay. Can you hear me okay, Lindsay?
Yeah, we can hear you.
Okay, I'm on the boat here. Great presentation, Scott. I mean, that's really impressive. And I truly do agree that they are very productive. I do have there are a couple things that come to mind when I think about the blue areas versus the red areas, or the areas that are, under the MPA. In that, how do you verify that the areas that you are using as the I'm not good with these words.
The reference sites?
Yeah, the reference sites.
Mm-hmm.
How do you assure that those are the same? Because I mean, we as anglers are very particular and have fished on the coastline for years and years and years, and we know where these creatures go at certain times during the year. How do you determine those reference sites?
Yeah, so the reference sites are determined through a collaborative process that involves talking with the fishermen and the fishing community and the sport fishing captains to try to choose locations that are as representative as possible. No habitat is completely, perfectly exactly the same. And so, but we really did our best, working with input from the angling community to try to choose those reference sites to be as representative as possible. On the Central Coast, we know that kind of the catch rates and everything started off pretty similar between the MPAs and reference sites. So that's a good sign that I think we did a pretty good job, right, choosing those places to be representative. There's also habitat maps. And so one of the analyses that we're going to start working on now is a real detailed analysis of the habitat inside the MPAs and the reference sites to see how much of the differences might be explained, by the habitat itself. And yeah, so it's a great question. I think it's one of the most important questions, because if they're not representative, then, you can't really compare.
Right. Okay. All right. Thank you very much, Scott.
Yeah. And luckily on the Central Coast too, right, we're able to see those abundances and the biomasses diverge over time. So that's a good sign that, at least they started off pretty similar, and you see those differences grow. So it really shows the value of the time series, right? And really understanding, kind of what's happening in these places.
Okay, thank you.
Yep.
Thanks, Dick. And I might encourage you to watch Rick Starr. His presentation, his webinar is the last one, and he focuses on fishing as well. Fishes and using deep water techniques, and they do assess habitat between reference and MPA sites as well. It might be really interesting for those that have that same question. We have a comment from Caitlin Gaffney. She said it would be great to hear from any volunteer anglers who might be on the webinar. So if you are one, just go ahead and identify yourself if we don't recognize you right off the bat. Our next question comes from Abigail. What were the approaches and methods used for volunteer angler outreach recruitment?
Yeah, so we use a variety of approaches. Some is, word of mouth from people that are participating in the program and getting their friends involved. Outreach from some of the sport fishing captains and sport fishing businesses that we work with. Posting on various, fishing blogs and stuff to get the word out. Yeah, those are kind of, I think, some of the primary methods that we use to try to get people involved. And we tend to get a lot of new people coming out every year. There are anglers that come out repeatedly, which is great, but we also try to get out new people every year. And so we get some of both. Yeah.
Fantastic. I see Jim Webb has his hand up. So go ahead, Jim.
Okay, thank you. Can you hear me okay?
Yep, we can hear you.
Yep.
Okay, I wanted to ask, excellent presentation. I thought that was really clear and really an accurate reflection of what's happened. I've participated with the CCFRP since the beginning, and we are really seeing some changes in MPAs versus reference sites. One thing that has concerned me is that the de facto seaward edge of state MPAs is at 3 miles because that's the limit of state's jurisdiction. And beyond there is a rockfish conservation area controlled by nymphs. And the proposed recreational fishing season next year for California shows that we're going to be able to fish at all depths from April to September. And that would mean that basically the RCA boundary is going to be removed. And I wondered if you had any opinions about what the effect of that might be on what we're seeing inside MPAs.
Yeah, that's a good question. I mean, those rockfish conservation areas have been closed since like 2002. So there's been these de facto large scale MPAs along the coast. Often they're in deeper waters, but some of the species, that we tend to catch with CCFRP are ones that, show up in the shallows when they're young, and then they move into those deeper waters as adults, where they then reproduce and make all the babies that then come in to the shallow waters. So I think we could see some impacts. It depends what the fishing mortality rates are, in the conservation areas. But potentially some of the reasons why we've seen some of these big increases, both in the MPAs and reference areas, are because there's been a large segment of the population that's been protected down there, that's been reproducing, making lots of offspring that have been augmenting those populations. So yeah, I mean, it remains to be seen, I guess, the level of fishing that's going to happen in those zones. And maybe Steve Words could speak to that and some of the regulation changes. But yeah, it certainly could have an impact because some of the species do move between the shallows and the deep, and they've likely benefited from those large scale closed areas over the last couple of decades.
Thank you.
Thanks for your question. The next one comes from Anupa. What are the avenues for outreach and process? Oh, we already kind of answered that question, didn't we? For recruiting volunteer anglers. That's the second of the same. Sorry about that. So Don on the line has asked, how are the MPAs enforced? And Steve Worts with the department has provided a reply in the chat, but I'll read it out loud really quickly. CDFW, California Department of Fish and Wildlife, routinely conducts patrols inside and outside of MPAs using a variety of assets, including enforcement partners. So if there's anything further to say, Steve, you can say it out loud, or I'll go to the next question.
Yeah, those boats sneak up on you quickly sometimes, come out of nowhere.
And there's some new news. Maybe I'll share it. So you didn't mention. You can find all the rules and regulations on CDFW's website. There are a variety of brochures, and those are being translated or have been translated and available online in a bunch of different languages now. So Spanish, simplified, and traditional Chinese, and a few other languages that are escaping me. So please feel free to go online and you can access those resources.
I'd also say enforcement can also happen from citizens right? Calling in or notifying people, because a lot of times I think people fish in MPAs not just because they don't really know the boundaries, and so just providing education can be really helpful. Or you can call into Fish and Wildlife and report, if someone's poaching illegally in an MPA, so that helps as well.
Yes, thanks for remembering that. There's a CalTIP line you can always call. The next question we have is from Oral or Nick. Where in your study is there a reference to the methodology in changing from barbless hooks versus standard hooks?
So we have a detailed, probably on our website, we have a detailed report that has all the methodology. It also talks about probably in the long-term Mpa monitoring report that we put together. But we, when we use hooks, they aren't barbless, but we crimp the barbs down to reduce hook damage. And so we've done that, I believe, throughout the whole life of the program as one method to reduce damage of the fish.
Great, thanks for clarifying. I don't see another hand raised at the moment, but we have a question from Dawn about offshore wind turbines, which perhaps maybe the department can touch on. If there are offshore wind turbines, how will they be handled in terms of MPAs?
Well, I think in general, all the proposed locations for the turbines are probably much further offshore into federal waters. But in some case, they may act as little MPAs themselves because fish will aggregate around structures, so they'll be attracted to those places, but you probably won't be able to get in super close to fish them. So in some ways, that may kind of add to some of the protected zones if they ever get put in, is my guess, further offshore.
Yep. Yeah, no, that's exactly right. I don't see any other new folks, but I see Digog just raised his hand again. So please feel free to ask another question, Dick.
Sorry, Lindsay. I just had a comment regarding the offshore wind and the cabling distribution and so on and so forth. I just got back from Tasmania, and it's interesting that they transmit electrical power from Tasmania to the mainland in Australia. What they found is that the bait fish and a number of the different fish themselves aggregate around the warmth of the cable. And so given these offshore wind turbines and the transmission lines that potentially could, will be coming onshore, it may be something to pay attention to because the overall impact could be very interesting in terms of the aggregation of the bait fish, the fish that feed on those bait fish, and the migrating species. They've noticed a difference in that area that I was in Tasmania. So just something to keep our eyes on over the long period of time here. Thank you.
Yeah, very interesting. Cool.
Yeah, thanks so much, Dick, for that comment. We have a few more questions coming in over the chat. Hayden starts off by saying that they've heard that time of day and tides can affect fishing conditions. How were tides and time of day factored into your Cpue of fisherman effort per hour? And have you seen that time of day or tides have an effect on your Cpue?
So certainly, the time of day, tides, the winds, kind of the sea state, a lot of that can have an impact on fishing. We try to deal with it by visiting each of these MPAs and reference sites multiple times in a year. And so it's not just one day a year, otherwise there might be too much variability. We don't really factor it in specifically to the calculation of catch per unit effort, but we do use some of these environmental conditions as covariates or other explanatory variables when we're statistically testing the factors that are affecting the responses of the MPAs. And in general, a lot of these sort of day-to-day variability just kind of comes out in the wash. So it adds to the error bars that we're seeing. But yeah, I mean, we're concerned about that, and we record information about it, and we include it in our analyses as a way to kind of control for some of that variability that happens from day to day.
Thanks, Scott.
Mm-hmm.
Abigail has a question, a good question in the chat, and I can take a stab at answering it from our end, and then I invite Scott, you to chime in as well. They ask, is your data or survey results publicly available? Rochelle has done a great job, and she's already dropped the links for CCFRP's long-term monitoring technical report, which you can find on California Sea Grant's website, on OPC's website, and linked to the department as well. And I didn't say it out loud, but there are these snapshot summaries, these 2-pagers that are distillations of the multiple hundreds of pages of amazing results that you can find in that full report as well.
And then, yeah, it looks like you just went to that page for the data. So the data is all publicly available. And maybe we'll go back to that in a second. Yeah, that's on the Data One website as our full data set, and it's updated every year. And so anyone can go in there and download the data and play around with it if they're interested. Sorry, you can go back to the report now if you wanted to do that.
Yeah, I encourage everybody to go read the full report. There's a ton of great information in there, and that can be accessed in a variety of ways. We have a few more minutes, about 4 more minutes for questions. So if you have a burning one, please drop it in the chat or raise your hand. If not, I have a quick one. It's a less sciencey question, but Scott, do you think you can talk a little bit about the multi-benefits of CCFRP? It's such a great program, bringing anglers, scientists, students together, and other ways that your science is used outside of MPAs.
Yeah, I mean, so for us, I mean, this program is so much fun, I think, because we get to work with, people in the community, all up and down the state. And not only do we have anglers come out to participate on the trips with us to help collect the data, but we also do these summaries every year to tell everyone kind of the results of the fishing season. We have these angler appreciation workshops that we do where we bring people in that volunteer with the program and show them the results of the season and get their feedback on how it went, what things we should change. And so we try to make it a very collaborative, involved and kind of transparent process. Yeah, our data is used a lot as well for fisheries management. So we work closely with NOAA NIMS and the stock assessment biologists. And many of these rockfish species in the near shore are considered to be data limited. And so we don't have a ton of information about the current status of those stocks. And so they're using information from our program to help inform the stock assessments for, so far they've done that for 7 rockfish species. Yeah.
Great. I don't see anything else coming through the chat yet, unless I've missed a hand. Ryan Fields just said, nice job, Scott, in the chat.
Ryan is one of my former students and a former technician for CCFRP now at Oregon Fish and Wildlife. Thanks, Ryan.
It's a great network CCFRP's built. Okay, here we have one from Oral. Were any fishermen involved when the tackle was selected for this study? As a longtime fisherman, I would not use any of the tackles selected.
Yes, there were lots of extensive discussions with fishermen, right, to try to come up with, the representative fishing tackle for each region. So I didn't really go over this in the methods, but we use different tackle. We use 3 different types. We use shrimp flies. We use shrimp flies with squid bait, and we use lingcod bars, usually with a little squid teaser on there as well. We do that in Central California. When we go down into Southern California, we keep it at every region. We keep at least the shrimp flies with bait as a standardized gear type. But in Southern California, the fishermen down there said, no, we don't use those same type of gear. We use swim baits and we use, circle hooks. And so we use those gear types in Southern California. We have a few modifications as well up in Northern California. So yeah, I mean, fisherman input has been really important to selecting the gear, but we needed to have at least one type that is standardized across all the regions so that we can easily compare our catches across all the regions using the same gear types. So yeah, we tried to balance all of those competing inputs for what the gear should be with some of the science that we needed to do.
Thanks for clarifying that. I know that's always, it can be a contentious issue.
Even amongst ourselves, every year we're still talking about the gear. So,
Oh, I bet. Yeah. Rochelle, I think there's a note here, but I don't see it in the chat. Is this something?
Yeah, so
you?
a couple of folks have sent me some direct messages, so I'm feeding
Got
on
it.
through there and through here.
Okay, thank you. So there's been some, let's say, collective comments. A lot of people expected that that inside an MPA, the fish would be larger and more abundant over time. But now we have pushed the fishery to congregate in the areas that's left, putting more pressure on the stocks. Shouldn't we rotate the opening of MPAs to take advantage of them? Tough question right at the end.
yeah, well, I mean, there's a lot of considerations, and in some places like Hawaii, they do rotational closures, and they can work, but often as soon as you open them, the abundance and the sizes, they get fished down pretty quickly. In California, I think, the goal of having these closed areas is to, protect some sections of the coastline. But there's some fisheries benefits as well by having these large fish that we've seen, larger fish and more fish inside the reserves. We know that larger fish produce exponentially more offspring than smaller fish. Those offspring that are produced, those larvae, leave these MPAs. They travel around for a couple of months on the currents and then show up somewhere else, often in a fished area, often helping to augment those areas. Right? The fish is also, yes, when they become more abundant in the reserves, densities can go up, but that's often when they start to spill over. If densities get too high, they'll spill over and leave the MPAs and go to areas where they can be caught by the fishery. So there can be some fisheries benefits for the reserves. And I think as well, if all the fishing effort was being concentrated outside the reserves and the reserves weren't providing any benefit, you would see that those catch rates, right, the sizes, the biomass would be going down over time outside the reserves, right, with more and more fishing pressure. But we don't actually see that. We see that the biomass and the sizes is either the same or it's also going up outside the reserves. So that's, I think, telling us that the fishing pressure isn't overwhelming those sites outside. So the MPAs are having some positive benefits in the fished areas, and likely we've had some good environmental conditions as well, right, that have kind of enhanced the populations both in and out of the reserves. But it's a good question.
Yeah, it's a great question. In our last few minutes, I'm going to turn it over to Steve with the department, but thank you all for your great questions and engagement. Steve is going to share a few notes about the decadal management review, and then we'll wrap it up after that.
Great, thanks, Lindsay. And once again, Scott, that was a wealth of information, and we really appreciate the effort CCFRP and all the program partners do to keep the program afloat and inform fisheries management, in addition to MPAs. So within the last few minutes we have here, I just wanted to briefly touch on why we're getting set up for this decadal management review and what will be involved in that process. So as you heard from Scott earlier, planning for the network was completed in 2012. And after that time, the state updated its master plan of marine protected areas from a siting and planning document to be a more forward visionary management document. So we established a 10-year management review cycle from the time the network was completed in 2012. And that took into account the life history and biology of the species that are found along the nearshore waters of California. And it's administratively feasible. So that was the reasoning for the 10-year cycle review. The department also, when it updated the master plan, established the Mpa Management Program, which is composed of 4 pillars shown in this slide here, and that's outreach and education, research and monitoring, enforcement, compliance, and policy and permitting. All of this information is going to be integrated that we've been collecting over the years into the first decadal management review. And if you could click to the next slide. So this slide basically represents the sorts and types of information that will be pulled together to report out to our Fish and Game Commission in February of 2023. Information like that Scott presented and the other Ask the Researchers, their information that they've collected over the last 10 years will be integrated into this review. We've been working with California Native American tribes to get an understanding of traditional ecological knowledge and their help and guidance on how they feel MPA should be managed. We worked with a wide range of stakeholders over the last couple of years, people like yourselves on this call who are interested in the marine environment. This type of information is all being pulled together to develop recommendations internally. We're working with our law enforcement. We had a question about that earlier. We're working with our fisheries staff. As another piece of the puzzle, we were fortunate over the last couple of years that the Ocean Protection Council funded several science advisory teams. One looked at the department's action plan and refined the performance objectives to be more effective in figuring out if the network is functioning as designed. We also had a group that looked at whether MPAs could be resilient to climate change. And more recently, they funded what's called NCES, the National Center for Ecological Assessment and Synthesis. And they're taking all the information I mentioned very briefly here, and they're looking across habitats and looking at the human dimension aspect to give us an idea of how the network is functioning. Is it meeting the goals of the Marine Life Protection Act? And with all that, we anticipate there'll be probably more gaps in our knowledge, more questions that need to be answered. But we're also hopeful that it'll inform us about future management strategies moving forward to be cost effective while getting the best bang for our buck and some really good adaptive management recommendations. So all that will come together. You can look for that report to be available in January of 2023, and we'll roll it out with our Fish and Game Commission in February. A subgroup of the commission called the Marine Resources Committee will meet in March, and they'll get into the weeds of all the information that we've produced. Well, not just us, all the partners, like the work Scott has done. And the day before that, I think it'll be like March 15th, there's going to be a symposium where we'll have all our partners who have represented the various pillars of the program, the researchers hopefully, there to discuss their findings, probably an open plenary session for public to ask questions. Hopefully we'll have, senators and maybe decision makers at that particular meeting, but that's in the future. But as we move closer to that date, we have our Decadal Management Review webpage, which is on the next slide. And if you go there, you can learn more about the process. Don't worry about writing any of these links down. They'll be posted in the chat. Also, if you have any questions about the Decadal Management Review, or if just MPAs in general, please send us an email. We try to respond within 48 hours. We always welcome questions. And so that's my 2-minute drill. I want to thank Lindsay and Scott for a great webinar, and I'll pass the mic to Lindsay now.
Great. Thanks, Steve. And again, thank you so much, everyone, for joining. It's always a pleasure to run these webinars, and I learn something new every time. Thank you so much, Dr. Hamilton, for your presentation. There's a lot of great information that's been shared in the chat, so please refer to that. There's links for the CDFW website, our OPC website. We're going to find a wealth of information. But most importantly, please take a look on the slide. You'll see that there's a flyer for our next webinar, which is actually this Wednesday from 5 to 6. We'll hear more about fishing from John Bukowski with EcoTrust and our Humboldt State partners. So with that, thank you for staying on, and we'll see you on Wednesday. Oh,
sorry, there's just one question about when recordings will be made available.
Oh, perfect. Thank you. Yeah, we will get this recording up within the next few weeks, and it'll be posted to OPC's website. The previous 4 are already available on our website. Hopefully that answered the question. Great.
I'll put that in the chat right now.
Thank you so much.